Alicia.
Perfect.
Yo voy a quitar mi cámara y mi micrófono para no interferir.
Muy bien. What a pleasure it is for me to introduce our speaker today, Professor Arturo Casadevall. He has a very extensive curriculum, and I will only summarize a few points. He is a Bloomberg Distinguished Professor in Molecular Microbiology, Immunology, and Infectious Diseases at Johns Hopkins School of Public Health. He received his training in New York and has more than 65,000 citations to his work. He is chief editor of the open access journal Bio and has been editor of many other journals. He has been president of several scientific societies, such as that of the ASM, American Society for Microbiology, and that of Mycology. Besides his over 700 publications, he is well known for his critical and novel ideas and enlightening concepts. But perhaps, Professor Casadevall, you didn't know that you are highly appreciated in Mexico. And thank you very much for accepting our invitation to participate. We may start today now. Just let me thank all the institutions that are connected virtually. Thank you very much, Professor Casadevall, again.
Muchas gracias, Esperanza. So I'll switch to English. I'm going to share my screen. Thank you for inviting me. I wish that I could be there with you. You know, we could have lunch and we could chat about a lot of things, but you know, this is a very, very unusual time, and we're living through historic times. And I think that the amazing thing is the extent to which we have keeping things going. And this is just an example of how we're trying to keep science and communications open. So I got to share my screen. I got to find my, oops, where is it? This one. I have it open, but I somehow don't see it. Hold on, let me try again. Maybe I should close some of these. Hold on a second. Let me close it and open it up again. Maybe that'll do it.
Okay, it's fine.
No. Show all windows. No, that's not. I don't know why it's not showing my, let me close everything. Maybe that's the thing to do.
Mario, que te mande la presentación, esa sería una solución.
See, yeah, but oh, I found it. See, that's there.
Okay.
So, now it is there.
Yeah, so what I needed to do was to close everything and then reopen it, and then it works. So for the next time, for the next Zoom catastrophe. So I'm going to talk to you about thoughts into the origin of microbial virulence. And this is going to be a big picture talk in which I want to pose to you a big picture question. So we're living through COVID. As we accumulate knowledge of all aspects of biology, how much of biology is going to be predictable? In particular, can you predict next problems in infectious diseases? But I'm also going to ask you some questions that are fun. For example, Why are we here? And the answer is not because to give a seminar. Why are we so hot? And by that I mean temperature. Why do we eat so much? And why are mammals the dominant large animals? And I'm going to give you the answer, which is a single word, and I promise you that this will all make sense in about 45 minutes. And the answer are the fungi. So I like to, we should focus on history. The germ theory of disease was one of those great moments in the history of humanity, in which things that were killing people for always in history were associated with individual microbes. And this was a revolution that happened in about 30 years in the middle to the end of the 19th century. I want to point out that Virchow did not believe in this revolution. So one of the great minds of the 19th century, the one who gave us the theory of cells, organ function, did not believe in this. And the reason for that is because he was very interested in social issues. And he felt that poverty was the cause of all these diseases. And if he accepted the germ theory, that what it would do is it would take away from the social causes. But I point out to you that as an issue because we live in a time of great polarization. And it's important to know that this is the way it's always been, and even great minds often don't agree. The germ theory of disease had a huge impact in mortality. What you can see is the rapid drop in mortality from infectious disease. Look at all the other causes. The blue line here remains about the same. I think when you extend this line, you're going to see a huge peak at 2020, which is the COVID. But the other thing that it did is it took away childhood mortality. And consequently, the population took off exponentially. If the germ theory of disease had not happened, a third of us would not be in this seminar today, simply because a third of us would not have made it out of childhood. But this idea that germs cause disease, there was a late 20th century crisis. So in 1900, there were two types of microbes, pathogens and non-pathogens. But in 1950, there began to be a problem. And that is organisms that were previously thought to be non-pathogenic began to be associated with disease. What happened in the 1950s, that's when they developed steroids, chemotherapy, antimicrobial drugs that affect the microbiome. And then the concept of opportunism came in to try to explain how non-pathogens cause disease. But this didn't really work because what they were trying to do is they were trying to define the microbe to give it an attribute that was not its own. No microbe causes disease without a host. And by the 1990s, if you had gone into a hospital, most of the pathogens in the hospital had been non-pathogenic 100 years earlier. So the question is, what happened? And this led to a crisis on how do we explain microbial pathogenesis. Whenever you see a messy lexicon, you know that there's a problem in the field. Let me give you an example. Candida albicans is a commensal, but it can be a primary pathogen for people with women with vaginal candidiasis or an opportunistic pathogen in AIDS. In fact, this organism can be a commensal and a pathogen at the same time on the same person. It could be in the skin in a commensal state, and it could be in another part of the body in a pathogenic state. And notice where I'm going to take you. I'm going to take you to the idea that you cannot define a microbe as a pathogen or a non-pathogen without taking into account the host. So in 1995, I was asked to teach a microbial pathogenesis course at Albert Einstein College of Medicine. I recruited my good friend, Lisanne Porofsky. And together we created our course in microbial pathogenesis. And one of the things we didn't want to do is, we didn't want to go to do the bug parade. What is the bug parade? You do the viruses, the bacteria, the parasites. We wanted to try to come up with an integrated view, and there was none. And what we realized was that our class notes had an integrated view. And then in 1999, we decided to publish it. The class notes was on it. So here is the problem. Today, thought is not valued. If you have a good idea, how do you publish it? That's very interesting. Where would Darwin publish today? He had no mechanism. You think, just to give you a sense of the problem, the intellectual problem in biology. So what you have to do is you have to put it into a mini review and you have to hide it in there. So this idea was first published 22 years ago, which was going to be the damage response to microbial pathogenesis. And it's a very simple idea, and that is that all host microbe interactions can be idealized by a parabola. And that the problem occurs, what matters is the damage to the host. And that this damage is maximized in weak immune responses. For example, the microbe can then take off, or in very strong immune responses, where the death is caused by the immune damage. In COVID today, what you see is that people start often with a weaker immune response, but then, because they have pre-existing conditions, but then their immune system is what kills them. Many people with COVID died with no virus on board. They die from the host response. And if you take that parabola and you drag it below the x-axis, then you could have symbiotic interactions. The important thing is that all the states are continuous. So this is actually quite revolutionary, because it basically says, don't focus on the organism, focus on the state of commensalism, the state of colonization, latency disease. So this view would have 5 consequences. It shifted the base from pathogen and non-pathogen to the outcome of the interaction. In our mind, there are no pathogens, there are no commensals, no opportunists, just microbes and hosts. You put them together and you get a new state. And that state can be commensalism, symbiosis, indifference. These states are continuous and differ only in the amount of damage incurred by the host. And virulence becomes an emergent property, and that would have enormous consequences for whether this will become a predictive science. So in 2015, we added the microbiome to this view. We have been updating it every couple of years, but notice that we still have to publish it as a mini review, because we don't have mechanisms for very good mechanism for publishing thought in the biomedical science. Now, one of the things that we realized is that the word hosts gets thrown around, but it had never really been defined. And we set out to define a host. So the question is a rock a host? No, I think people would say no. And the reason for that is because it doesn't respond. To respond, you need to have an immune system. You need to have something that can create a new state. And that new state will be the state of symbiosis, colonization, commensalism, latency, or disease. So after a while and many years of working on this, Nature approached us to write about this. And we wrote this provocative article called Ditch the Term Pathogen. The reason for that is when you think, you think using words. What happens if your words are not precise? What I would argue is that the thoughts are not going to be precise. And the term pathogen is a very problematic term because it gives a microbe a term, a quality that is not its own. Give you an example, smallpox. Most of you are young, so you are susceptible to smallpox. I'm old enough to have been vaccinated. So it doesn't matter, a variola virus, how many virulence traits it has, it is not a pathogen in me, simply because I'm immunized. So it gives you the problem of definition, that you cannot define these entities from one view alone. So one of the things I'm very interested in mathematics, as you want to see, and I wanted to know, could a mathematical formalism be developed to do this? And playing on my own, try to develop the pathogenic potential of a microbe. Can you come up with a simple formula that anybody can use that will show you that this is in fact the case? And I found something that works. It's called the pathogenic potential. You look at the fraction symptomatic over the inoculum. The inoculum, it gives you the microbial factors. And then you multiply it by a term that takes into account mortality, because mortality is something that is very profound. The host dies. So if there is no mortality, for example, if all your mice only lose weight, the n becomes 0 and this term becomes 1. And then I went into the literature and I basically decided to just, this is for mice, found papers. And what you can see is that the theoretical maximum is 10. The next one is Francisella tularensis for mice, in which is 2.5. but everything is continuous. There is no break. There is no place in which you're going to say, that's not a pathogen. The reason for that is because you can always kill a host with increasing the inoculum. You can also now do the same experiment, go to an immunology journal and look for papers on a mouse strain in which what you can calculate, and you can see that as you change the host, the pathogenic potential can change 100-fold. So here is a formula that works. So instead of talking about SARS-CoV-2 is a pathogen, what you can say is SARS-CoV-2 has a pathogenic potential that can in fact be calculated and can then be compared to SARS-1 or can be compared to MERS. This is beginning to be used, and it gets you away from things like virulence. Virulence is a problematic property because it's a microbial property that is dependent on a host. Therefore, is not an independent microbial property. But you can calculate the pathogenic potential, and it gets you away from some of these very difficult problems. So I'm going to now go to the fungal world, because it teaches us some lessons on the problem of virulence. Now, I work on bacteria, and if you look me in the last year in PubMed, you would think that I work on COVID. No, I'm interested in these problems. I'm interested in learning from wherever things can come. So the fungal kingdom is, I'm using old terminology here, is probably the largest kingdom. It's got millions of species. It's the major pathogens of plants, insects, invertebrates, and ectothermic vertebrates. Those are cold-blooded ectomers. Fungi are currently devastating major ecosystems. The bats are being devastated by the white nose syndrome in North America. We have had amphibian declines, the salamanders declines in Europe, snakes in North America. But most people don't worry about fungal disease. Why? Mammals are remarkably resistant. So what makes mammals so different? In fact, there are very few fungal species that are pathogenic for humans, very few, where you may have hundreds of bacterial species. In the fungi, there are no more than 5 or 6. And there's some are host associated like Candida, in which you have disease when you have a breakdown of the host microbe relationship. And then there are a few environmental organisms, of which these are the major ones. So fungi, as I said, are devastating entire ecosystems. When the fungi get into an ecosystem, they often kill every last member of species. And then they go back to living in the soil. These are not like viruses. These don't require the host. Don't need a host to maintain themselves. They can kill all the frogs in a pond, and then they just go and live in the water. So I'm going to talk primarily about Cryptococcus neoformans because that's the organism that I work with. We are all infected, especially if you live in cities. But so infection is very common. Disease is rare, and disease tends to occur only or primarily when you get a very large inoculum or one becomes immunosuppressed. This is an organism that has well-defined virulence factors. It's got a capsule, and it makes melanin. And in fact, we have been able to estimate that these two traits comprise about half of the total virulence composite for these organisms. The melanin is deposited in the cell wall and protects it against everything from ultraviolet light to things like enzymatic degradation, defensins, even heavy metals, and protects it against antifungal agents. So here is a conundrum for you. It has a very sophisticated intracellular strategy, but it doesn't need an animal. And it can cause disease in many different hosts. So think about it, HIV has a sophisticated strategy, but it only causes disease or infection in humans. This organism can kill a fly, this organism can kill a mammal, this organism can kill a worm or a ham. So the first question is, why does a soil organism with no need for an animal host have a sophisticated virulence strategy? Second question is, how does a soil organism with no need for any host manages to suppress such diverse defenses? So the first answer, we got a hint 2 decades ago, and that is all this virulence strategy is designed to survive amoeba. So this organism lives in soils. In soils, they are preyed by many species of amoeba. The capsule, the melanin, the enzymes are all defense mechanisms that then function in virulence once it gets into your lung. So the capacity for virulence is accidental. And this has now been found with many other soil type of organisms. The next question is, how can you take down such very different eukaryotic hosts? And the answer is, it targets common pathways. So this is unpublished work, but this is the work of Carolina Coelho. And what she showed is that, think about it, if you're going to take on an amoeba, or if you're going to take on a worm, or if you're going to take on a mammal or a plant, what you want to find is the common pathways. So it tends to target mitochondria and the ribosome genes, which are very conserved across all eukarya. And the reason for that is simple. This organism is sitting in the soil. It doesn't know who's going to eat what organism is going to eat it next. So what it does is it needs to have a strategy to survive, and it does so by undermining key pathways in these organisms. We also know that this strategy is ancient. It goes back to at least the Cretaceous. How do you know that? Well, what you do is you get strains that are very different, very distant, and then you study them in detail. And what you can find is that even though they separated 100 million years ago, they do the same thing to a macrophage. So what that's telling you is that this strategy has been conserved and it's been successful. And probably it is because it targets common pathways. In fact, we have been able to think, we know that the organism originated in Pangaea, in the Cretaceous, and it probably originated in an area where South America and Africa now used to touch. And we have proposed that with continental drift, what you had is that the speciation follows continental drift. And in fact, other people who know more than I do have taken this map and this idea and been able to plot the different lineages. And what you see is that this now has a lot of explanatory power. Now we understand why some of the Spanish isolates are very close to Eurasian isolates, because they were very close in the distant past. So pathogenic strategy goes back to the Cretaceous, probably diverged with continental drift. Why is this organism pathogenic? Well, one of the things, we're going to ask the question a different way. Why are the mammals so resistant? So the work of John Perfect and others provides an answer. So bunnies are very resistant to cryptococcal infection. You cannot give them systemic infection, even if you shoot it directly into the brain. Instead, you can give them a skin infection, and the answer to that is because this is cooler. But if you give steroids, heat is not enough, and you get a dead bunny. So what you're seeing here is that the emerging pattern is that this resistance is coming from two areas: high immunity, that is adaptive immunity, having B and T cells, and having being warmer. An example of this is found in the white nose syndrome in bats. So this is a problem in North America that began in 2006, when this organism was probably introduced from Europe in a cave. So the bats are resistant in the summer. In the summer, they are 37 degrees, but in the winter, this fungus grows on them when their temperature drops to about 10 to 12 degrees. And what happens is the white nose is because they get this hyphal growth around the nose. If you take the bats into the laboratory and you just feed them, they rapidly raise their temperature to 37 and they clear the infection. But in the middle of winter, they can't because they don't have food. They are hibernating, and that's how they die. So one of the things that we did is I work with Vincent Robert, and we asked the question, what is the thermal susceptibility of the fungi? And he has access to a culture collection in the Netherlands that had over thousands of isolates. And what you can see is the data on the top. It's fungi are environmental organisms, so they do very well to about 30 degrees. But their ability to replicate drops dramatically after 30 degrees, such that by the time you reach mammalian temperatures, there are very few that can replicate. So Aviv Bergman, who is a friend and mathematician, I told him about this problem. And what he decided to do was to take the temperature, to take the cost of being hot and the benefit. So the cost of being hot is how many calories do you need to eat? And the benefit is protection against fungal diseases. And then to solve these differential equations and ask, where is the maximum? And remarkably, the maximum is 37 degrees. I'm sorry, this is in Kelvin, this thing, but this graph should be 37 degrees. So that suggests that, gee, maybe our temperature, mammalian temperatures, are optimally designed to keep out the fungi. So with the amount of heat that you need, that is, if you could always be 42 degrees, but then you may have to eat 6 times a day, because that's why, incidentally, why we lose weight so rapidly when people have febrile diseases. These relationships are not linear. You need to eat a lot more if you go up just a couple of degrees. The cost of the mammalian lifestyle is very expensive. This is a log plot. And the elephant, us, the amount of energy that we need to get through the day is enormous compared to an organism that doesn't regulate their temperature. So one of the questions that has bothered me over the years is, how does this lifestyle that requires so much energy become dominant? Why are mammals the predominant large animals? Mammals and birds, they're both pretty hot, and they both have tremendous heat. So one of the things is, could the explanation be in the distant past when the mammals emerged? So if you look at the history of the Earth, of the biota, the protozoa and the fungi go way back. Land animals only appear about 450 million years ago. For most of this time, it's the age of the reptiles. And then something happened about 65 million years ago. The dinosaur lines died out, the birds survived. But what you see is that it's in the tertiary, our time, the large animals tend to be warm-blooded. And this is a huge change in the energetics of the animal kingdom. So how do we get here? So if you ask the question, what killed the dinosaurs, you'd be surprised at how many answers people have proposed over the years. Some people used to think that the mammals ate the eggs. That doesn't make any sense. But anyway, that was a mammalian-centric idea that you could find in the early literature. I found this in the internet. I don't think this applies either, but it's funny. A massive volcanism may have contributed. 65 million years ago, the Deccan Traps. This is a magma plume that opens up every now and then. It created the Hawaiian Islands, for example, opened up and spewed a tremendous amount of volcanic gases. And then within 100,000 years, an asteroid hit in the Yucatan. And this is the calamity that is thought to have really killed the dinosaurs. There are other ideas out there. I'll share some of them with you. This was in a Cell Press journal. You can think whatever you want of this, but some people calculated that they could have set up a global warming by passing gas. And this is something that I don't have any data for or against. So I'll show it to you in silence, since I can't comment on it. So the end of the Cretaceous, the Earth was a warm, volcanic, and forested planet. There were forests in Antarctica. The Atlantic was tiny. and the world was dominated by the reptilia megafauna. The asteroid hit, as you know, the Yucatan. And one of the very interesting things is that the ring of cenotes is the rim of the crater. And the reason for that is because the limestone melted and cooled very rapidly and created those geologic formations that are found no other place in the world. So the planet had a really, really bad day. The tsunamis reach into most of North and South America. The fires and dust led to global cooling of about 10 to 12 degrees over 1 or 2 days. The fires were started because a lot of Earth gets kicked into low orbit, and those rocks now entered as flaming meteorites, and you have dying forests because there is no sunlight. So there is very good evidence for this cataclysm in the geologic record. There is also evidence for massive fungal proliferation at the KT boundary. Think about it. The world is warm, it's cool now, dark, and with massive amounts of dying vegetation. It's the perfect compost. So what would have happened to the survivors? The survivors would have experienced large numbers of fungal spores. And one of the things from the pathogenic potential is that even low pathogenicity organisms can cause disease if you increase the inoculum sufficiently. However, there was one class of animal in this entire that was warm-blooded, and those were probably some of the early birds and the mammals. And these would have had a huge advantage because their temperature would have kept out the fungi, but also they would have had the advantage that they could move. Reptiles can't move very well when the temperature drops and in cooler temperatures. They can't even digest their food. So what you have is a fungal filter. I have proposed that a fungal filter at the KT boundary selected for the mammals. This was first written down about 16 years ago, and it's been updated through the years. So the post-impact world, the reptilian megafauna was killed by the impact blast, almost certainly by the disruption in food sources, the chilling of the planet. Maybe fungal diseases killed off some of the survivors. Reptiles fight off fungal infections with induced fevers. They find a hot rock and they kill off the fungus, but there was no sun. Reptilian eggs are susceptible to fungal diseases from the outside. In fact, you can find dinosaur eggs invaded with fungi. You can still see the hyphas. So, but you know, we still have some reptiles today. So some reptiles did survive. The question that always bothered me is, if you began with some reptiles and some mammals, why did the mammals win out? Well, when they needed so much energy. Well, to me, it's that temperature. That temperature allows them to survive the calamity and to survive fungal diseases. So the way to I think about this is you had the cataclysm, a global compost then resulted in massive amounts of spores and selected for endotherm. So now this is internally consistent. Mammals don't get fungal diseases because they were selected by the fungi. And this is why fungal diseases are so rare among warm-blooded organisms. This has been updated a few times. This is from a paper that was published earlier this year. So what you see is you have the impact, tsunamis, and the decline of the ectotherms through a mushroom and fungal bloom. The mammalian ancestor then already had advanced immunity, was a vertebrate, and then went through these bottleneck and came out with the great radiation because it was much more fit than the reptiles. So the view is, I gave this talk at the NIH and somebody made this poster for me. So you have the great reptilian megafauna, fungal selection creates a filter for the emergence of mammals, and you have the mammalian megafauna. So what are the requirements for fungal human pathogenicity? Well, you got to be able to grow at 37 degrees, but only a few organisms do that. But you also then you need the virulence factors. You need to have the traits that allow it to survive in the host. So the combination of the two gives pathogenic potential. And even with pathogenic potential, the majority of the fungi cannot cause disease in immunological competent hosts. So we are now facing, however, climate warming. And if you look at fungal diseases, they tend to be primarily near the equator. Why? It stands to reason that organisms near the equator and the warm regions are more likely to have adapted to higher temperatures. But one of the things is that fungi can rapidly adapt to temperature. Here is a paper from BMC Biotechnology where they, within 6 months, were able to adapt a fungal pathogen of insects to grow at 37 degrees. Why was that important? Because if you want to use it as a pest control, the insects sun themselves and kill off the fungus. So they wanted to develop more that was thermally stable. And we have warned that with global warming will bring new fungal diseases simply because the fungi will adapt. And many of them that currently cannot grow at 37 degrees, for example, the Tmax maybe 35 degrees, they're already fully loaded to cause disease. If they adapt to 37 degrees, 38, 39, they are then able to break through our thermal barriers. Now, Candida auris is a new disease that appeared about 10 years ago simultaneously in 3 continents. And it's resistant to drugs. And the question is, where did this come from? It emerged simultaneously in Venezuela, South Africa, and India. And we have proposed that this is the first example of a fungus that has adapted and has broken through the thermal defenses. So if you think about resistance to fungal disease, there are 2 pillars. You have advanced immunity. If you break this pillar with HIV infection, cancer, and drugs, you get fungal diseases. Now, endothermy is very difficult to break, right? Because we don't have any clinical condition where people are cooler for a very long time. But you could defeat it by making the organisms more thermal tolerant, such that they're no longer inhibited by your temperature. So I think that both pillars are under threat by modern medicine, by AIDS, by other infectious diseases, and by climate change. We have evidence that the fungi are already adapting. So what you see there is a very noisy curves. They come from the average temperatures at which isolates are deposited in a collection. Think about it. You send an isolate to a collection. What they often do is they study it before they put it out. They do the temperature susceptibility, what carbons it can grow in, etc. And what you can see is that there are 4 lines here. The 2 top ones are the ascomyces. These tend to be thermally adapted already. They are not changing. They provide a control. But the basidiomyces tend to have lower thermal susceptibility. And beginning in 1985, they seem to have been trending upwards. Evidence that the fungi are already adapting. So the question that I now pose to you in the last 15 minutes or so is, can we predict future threats? So if you think about epistemology, let's talk about our knowledge of our solar system. First, you had people that look at the stars, stargazers, and then they saw patterns in the stars. And then you have theories of the cosmos. You had the Ptolemaic theory that lasted for 1,500 years. And then you had the Copernicus theory. One posed the Earth at the center of the universe. The other one put the sun at the center of the solar system. And then you had mechanism. But there is a deeper knowledge, which is the mathematics of the system, the dynamics of the system. So when people say they want to do mechanistic science, what they often say is, I want to do deeper work. But you can see these are the levels in epistemology. And if in infectious diseases, you can have them too. You have observation. Is the host susceptible? You can look for patterns. things that are more, you can do experiments to see what is more virulent and less virulent. We now have a theory, which is the damage framework that I told you about. Many people work on mechanisms. They want to understand how virulence factors do, but very little work has been done on what is the mathematics of the system. Now, things can be deterministic. That is, if you know everything, you can predict into the future. They could be chaotic, which is a form of determinism, as I'm about to tell you, or they could be random, in which you would not know in any way what the future would bring. So in one slide, I'm giving you an entire field of mathematics, which is the dynamical systems. What you need to know is that there are only two major ways a system is dynamic. It could be deterministic. That is, if you know all the conditions and there is no randomness, you know the future of the system. Chaos in mathematics is deterministic. The problem is that chaotic systems, very small changes, propagate, as I'm about to tell you. But chaos in mathematics doesn't mean chaos in daily life. Or they can be stochastic, in which there is no prediction. The best example I can say, you can suspend an atom of carbon-14 in a beam. You don't know, no theory can tell you when it's going to decay. It can decay in the next minute, or it can decay at the end of time. That radioactive decay is completely random. No power in the universe can explain that. So moving forward, let's talk a little bit about chaos. Chaos is this view that the system is deterministic. If you can go back to January 5th, 1985, and set up the weather the way it was then, the rest of time plays out the same way. But the bottom line is you can't, because very small changes in recreating that weather propagate through time to give you completely different outcomes. This is why some people have used the argument, a butterfly beats the wings in South America and creates a tornado in Texas. So detecting chaos is mathematically actually difficult. You need to do what are two types of analysis. One of them is an ordinal pattern analysis, and the other one is a power analysis. And what you're looking for are forbidden patterns. Imagine, right, if you have a system that is driven by chance, you should be able to find all the possible states. But if you're missing some of those states, it tells you that is deterministic. And then you do what is called a power spectrum analysis, in which you ask the question, is it periodic or chaotic? Very complex math. I'm not going to take you through this. I'm going to try to give you results. So here is an example of what you look for. You analyze the data, and if it ever goes to zero, that's telling you that it is deterministic. Doesn't tell you whether it's chaotic or periodicity. But if it doesn't reach the bottom, then it is stochastic. So let's look at something like chance in the phagolysosome. So we began to look at something as simple as acidification of a phagosome. So the cryptococcus is taken up into a phagosome, and you can very nicely measure the pH by looking at the ratio of fluorescence. And what we found was that when we analyze all the pH intervals, this is tens of thousands of them, we could see in no case you see the curve goes to the bottom. What that's telling you is this is not a deterministic system. So we already know a lot. We know that this system is driven by chance. And that means it's going to be unpredictable. You will never be able to predict what a phagosome really pH goes to. That chance is probably set up by the number of pumps that are recruited to the phagosome. Now we began to analyze what do cryptococcus do to that phagosome? Is this a defense mechanism of macrophages? So what you look is that if you just put beads in, it's a beautiful normal distribution. In fact, most of the measurements, except at 3 hours, there was a little deviation from normality. Beads can change the phagosome, latex beads. So everything that is happening as a phagosome is formed, the pH that it's going to have at the end is going to be a stochastic, part of a normal distribution. What happens if you throw Cryptococcus in there? Oops, it's no longer normal. Cryptococcus changes the pH away, the distribution away from normality. So this is what you see on the right hand. By the way, this has been published in the Journal of Clinical Investigation last year, if you're more interested in this. But what I'm giving you a big picture thing. The Cryptococcus forces the distribution away from normality. The macrophage wants to be normal. The Cryptococcus wants it away from normality. What are the mechanisms that it uses? Well, it secretes urease that hydrolyzes urea into ammonia, raises the pH. The capsule has a large capsule. If you look at the volume, it dilutes the inside of the phagosomes. It secretes phospholipase that damages the phagosomal membrane. So you could have stuff leaking from the cytoplasm. And the capsule has glucuronic groups that buffer the pH. So what you have is, this is an organism that is used to be eaten by amoeba. It basically knows that whatever the amoeba is going to do, you want to mess with it. And survival means messing with the physiology of the phagosome. So what we have here is that the macrophage is using the normal distribution as a bed hedging strategy. When a macrophage eats a microbe, it doesn't know what the pH susceptibility of the microbe is. Think about it. You could be, so you hedge your beds by putting that phagosome in a continuum, where the majority of them are going to be better off in inhibiting the microbe. Bed hedging is very common in biology. Look at those eggs at the bottom. Now, the little egg would be better likely to survive in a bad year. So animals, what they do is they hedge their beds by generating diversity, because one of those large eggs is going to be very high fitness if it is a good year with a lot of food. But if it happens to be a bad year, well, the one that is smaller could do better. So here we have analyzed this, and what we found is that the macrophage is hedging its beds by basically generating a pH distribution that inhibits most of the things that it eats. In this case, Cryptococcus, but things like tuberculosis, Candida, a lot of other microbes want to move away from normality, because by doing so, they put themselves in a place in which they have increased fitness within a macrophage. So Monica Garcia Solache was from Mexico. She was a postdoc with me for a couple of years, and she took an early peek at chaos. She did a lot of work, and she did not find it in a cryptococcal larva interaction. Now, it's very important that a negative result, not finding it, doesn't mean it didn't exist. We thought that the system had to be chaotic, but she didn't find it. And the question was, do we needed to do the experiments differently? Well, Aviv Bergman, and we have continued to work, and this is a paper that is in preparation, is unpublished. We have now looked at 3 very different systems. Nicole Brother looked at Drosophila, we looked at C. elegans, and we looked at Galeria. And in the Galeria experiments, as well as in C. elegans, look, you see forbidden intervals. The analysis takes you down to 0. This is telling you that the system is deterministic. This is a signature of chaos in infectious disease. If host microbe interactions are chaotic, that means that they're not going to be very easily predictable. In fact, it's going to be very hard to predict. So on prediction, it's going to be very difficult. Virulence factors manifest new emerging properties that could not easily be predicted from their known roles. I just gave you an example, urease. It's used by the Cryptococcus for getting food in the soil, but it one of its product is ammonia. So one of the things that it does is it generates ammonia that affects the pH. That would have been very difficult to predict from first principles. Host cell microbial interaction in the phagosome are stochastic, implying randomness in final outcomes. And then you have chaotic signatures. So here you have all the elements for uncertainty. You have randomness, and even though chaos is deterministic, one of the things that it makes it chaos is that because the initial conditions are so susceptible to small changes, you can essentially becomes irreproducible. You cannot reproduce the initial conditions. But I would say don't despair, because weather is the ultimate chaotic system. And with computers and modeling, weather forecasting is getting better. Let's look at these curves over here. 25 years ago, a 10-day forecast had a probability of being right about 30%. Today, a 10-day forecast is approaching 50%. This is all done with modeling and computers and feeding in data of the history of the system. So what I'm getting at is that I don't believe that host microbe interactions are going to be ever predictable. You can recover many coronaviruses, you can study them, but which one is going to give you another COVID situation? You may be able to issue a probability forecast. This one has an 80% probability, but I don't believe it's ever going to be deterministic to the point that you know so much about the host and so much about the microbe that it will be predictive. I want to end with what my lab did. So my lab took this mathematics and began to analyze my visits to the lab. So what they did is they began to look the intervals between me visiting the lab from my office during an entire summer. The entire lab cooperated with this without telling me. And what you can see is that in my life, there are some chaotic generating events because there are at least 3 forbidden intervals. Now, I was so impressed with them and so happy that they had done this. But then when they presented this to me at lab meeting, I immediately became horrified because they had done human research without IRB approval. So I immediately called up the dean's office and I said, listen, I don't care what they did, but this is so creative. And it shows you what you can do with mathematics. For example, the people logging in and logging out of this seminar, you can analyze the dynamics of that as a chaotic, random. And so the dean said to me, don't worry about it as long as I don't publish it. It's not research. So I told them that obviously we're not going to publish my analysis of my lab visit to the lab, but it shows you the power of thinking this way, that there are patterns beyond our senses, and that those patterns could be very informative to what we do, and that may be the deepest form of knowledge. In the solar system, for example, we know that Pluto has a 30% probability, Mercury, of being kicked out of the solar system. We don't know when, we don't know if it would happen. But that level of prediction, we can predict a lot of things, but we may not quite predict the fate of the planets with time, because the system is fundamentally chaotic. So I want to return to the question that I posed to you earlier. Why are we here? The KT event of fungal selection. Why are we so hot? To keep the fungi away. Why do we eat so much? To maintain high temperature. Why are mammals the dominant animals? Fungal selection kept down the reptiles. There was no second nature to the reptiles. So remember I said to you, why are we here? Why are we so hot? Why do we eat so much? Why are the mammals the dominant animals? And I gave you one word, fungi. I believe the fungi are critical to answering this question. So I just want to acknowledge the many people who worked on this. This is an old photograph, but some of the key players are represented there. I try to give credit to everyone by showing their pictures when the work was presented. So with that, I'm going to stop sharing And I'm going to thank you for having invited me and for listening to me.
Thank you very much. We are open for questions now.
So I think the people can handle the hand to show in the chat from the participants. Sorry.
Arturo, even though mammals are not easily made themselves sick with fungal infections, they are susceptible to the toxins of to fungal toxins, and they are to make mutants, and they can kill them. So an explosion of fungi in KT would have affected mammals as well, no?
Yes, it would have affected mammals as well, but it's all relative susceptibility. If you if a lizard, for example, would have been in much greater difficulty than a mouse at that time, simply because they both would have been affected, but the higher temperature of the mouse would have at least prevented systemic infection.
But mammals have to eat something, and then if they are infected with fungi, plants, or the lizards.
So it turns out that that many of the small mammals eat fungus. And one of the things that happens is that if you think about
11 horas.
the KT boundary, that it was dark for 10 years. There was no photosynthesis, but there probably were lots of mushrooms. And organisms that were warm would have been able to move and eat, whereas organisms that are ectothermic can't even move to look for food.
Okay.
So I have a question.
Sure.
So this host vector interaction or is only with microbes, this chaotic behavior can be applied to larger parasites, for multicellular parasites, for instance,
worms. Yes, I believe that this is fundamental. I believe that you have two entities, and they interacting, whether it's a worm or whether it is a virus, that these are very foundational issues. So we did it with 3 different organisms, but I suspect that if you set up a system with helminths, or if you set it up with coronavirus, the result would have been very, very, very similar.
Yes, very similar.
Yeah.
Okay, thank you.
I think the important thing is to think about the limits of predictability. Especially, there are now plans to begin to set up sentinel programs to look for viruses out there. There are supposed to be at least 200,000 mammalian viruses. How would you predict? How would you generate a threat scheme? And I think the message is, yes, go on and do it. But even the best of it is probabilistic. And that is a limitation on what you can know, an epistemic limitation in the system. So I have a question here from Isaac Olivar. So it is likely that the higher prevalence of fungal diseases in humans in tropical and subtropical regions is due at least to adaptation of fungi to warmer environment temperatures. So the way to think about it is, you got fungi all throughout the planet, including in the Arctic and in Antarctic. But any fungus that is living near the equator has to experience really hot days. And those fungi are more adapted to higher temperatures. So if you happen to then be more adapted to higher temperatures and also have the virulence factors, that organism statistically is more likely to be able to cause disease in mammals. And this is why we think that most fungal diseases are in the equatorial regions. So, hello, thanks for this interesting and provocative talk. Shall we rethink the term virulence factor? Yes, I think it's a garbage term. What is a virulence factor? A virulence factor is something that confers on a microbe some ability to cause disease. So it can be a capsule, it can be a toxin, it can be a protease, it can be an enzyme. The problem is that we don't think about these things globally enough. For many of these organisms, what is a virulence factor is nothing more than a tool. For example, in the fungi, the proteases are used to obtain food. So when they get secreted in tissue and they destroy a tissue, it's not because the fungus they were designed to cause disease. It is just happened to be accidental. So I do think that we need to look at this more globally and more questioning than to think that these things were designed for virulence. They're designed for other things, and they just happen to work accidentally. But if you mutate these factors, they die when inside the host. Absolutely. If you have pneumococcus as a capsule, you take the capsule away, the capsule doesn't protect it. They do have a role in survival inside the host, but it is important not to think only in terms of these things are designed for causing disease, but rather that they may have other roles in the physiology of the microbe. What about viruses? Well, viruses are different in that viruses are completely dependent. Sorry, there's a cat bothering me here. Here's the cat. This is what happens when you do Zoom from home. Viruses are completely dependent on the cells to replicate. So they're not free living. So they do have virulence factors in the sense that some of their genes are often needed for causing disease, either by triggering inflammation. But one cannot separate them from their hosts because they need the host in order to replicate. Hello, thank you for the presentation. I didn't understand why fungi are so effective in their pathogenicity. Fungi are not that effective in pathogenicity. They are effective only for organisms like plants, not for mammals. Mammals, most of the people in this conversation do not worry about getting a fungal disease. I think that they happen to be eukaryotes. They happen to have a lot of very different metabolic systems. Fungi make a lot of secondary metabolites, and this is why they can be so effective pathogens for plants. They can be effective pathogens for insects. The mammals are not the case, probably because of our temperature, and I would argue because we were selected for the fungi. Thank you, Dr. Casadevall. I once read an article by you about the emergence of Candida auris and climate change. In our laboratory, we have worked with Candida humulini. It's not capable of growing at 40 degrees, but in blood agar, it does grow at 40 degrees. What does that say? When it is said that the fungus is unable to grow at 37, shouldn't different culture media? Absolutely. I think that that's a really important point. I think that carbon sources make a huge difference in metabolism. And you can imagine that if you're growing on YPD or SAB or blood agar plates, the metabolism of the organism is different. So the temperature susceptibility is going to be different. I did not know that. I think those results that you mentioned there are very, very interesting.
So there's another question.
You mentioned that there are some studies on fungi that affect mammals. Does this interfere with generalizing that mammals are resistant to fungi? No, the generalization is simple. I ask yourselves, how many of you worry about dying of a systemic fungal disease? You worry about nail fungi. You worry about maybe athlete's foot. But in general, fungal diseases don't tend to kill intact mammals. Now, some of them do, but you have to look at the bigger picture. We are, however, quite susceptible to bacteria, susceptible to parasites, susceptible to viruses. And the question that to me has been very interesting is, why this resistance? What makes us so special? And the data that I presented today and the arguments is that this specialness comes from having two things: adaptive immunity, which all vertebrates have, but also having higher temperatures.
So I think we are going to take the last question because we have to move on for the class. So I think the Susana Lopez question, probably the last one, and probably you can ask, all of you can ask Dr. Arturo to directly to his email or something like that for different questions, or to review the chat later for him.
Okay.
So then the last question is from Susana Lopez, I think. You think that fungi are predators for excellence and they are driving evolution, right? But the viruses can also predate fungi.
Oh yeah. All these things have been ancient. Probably, you know, the first event that led to a eukaryotic cell was probably an infection event. I mean, think about how organelles came to be and things like that.
Okay, so then we are going to stop this talk, the presentation. Thank you very much for a very nice and interesting seminar. Now I learned a lot about the evolution of mammals, probably, and the successors of mammals. I never thought about fungi, but well, now I have to think about that. And thank you very much.
Thank you, Mario.
And thank you to all the people to assist to this talk by internet. And now we are going to continue with the class. So then the students have to reconnect, to maintain the connection. And then